Hello everyone, good evening to all of you. Thanks so much again for tuning in. We are back from Thanksgiving/fall break at my campus and my college, so that means we have to go back and do more notes and get back into the swing of things, especially microbio. Alright, so this set of notes from the professor was over antimicrobials as well as antibiotics, all those good things. Alright, so a little history on the chemotherapeutic drug. AKA any chemical that is used to treat a disease. So in the history of chemotherapeutic drugs, we have two main players. We have a scientist named Paul Ehrlich, who'll talk about them as magic bullets, wherein the magic bullets would only harm these horrible microbes that are causing diseases, so they would only harm the pathogens and not the hosts. For example, one of them at the time was salvarsan for syphilis. So at the time, that was known as a magic bullet because it only affected the microbe that was causing that disease. We also have another scientist named Domach. Sorry about that for the pronunciation there. But this scientist, Gerhard Domach, he noticed that the red dye, known as prontosil, could be used for strep infections. Therefore, from all that, we do get the terms used nowadays, such as antimicrobial, which is any drug regardless of origin. Any antimicrobial drug, I should say, regardless of origin. And then we also have antibiotic. And this is going to be the substance made by a microbe to inhibit other microbes. Let's highlight these vocab terms. Okay, so again, when we're talking about antibiotics, we can't ignore Alexander Fleming, who discovered penicillin. More on salvarsan a bit. There's a danger to using this magic bullet, this drug, because in the molecule of salvarsan, we have some arsenic, so that causes some rashes, some bladder, some kidney, and some liver damage as well. Right, more on Fleming. He's going to be working from 1881 to 1955. That's when he was alive. He's a bacteriologist and a doctor. At St. Mary's in London from, again, 1881 to 1955. He's going to find the penicillin drug made by penicillium mold. And there's a specific type of penicillium that actually makes it. We'll get to that in a bit. So, early antibiotics in 1940, there's going to be the first clinical trials of penicillin performed by Florian Ernst Chain. And in 1941, there's going to be, again, the first clinical trials, and then there's also going to be a treatment of a police officer with a life-threatening staphylococcus infection. Now, we move our penicillin production to the US during World War II. And that's so this medicine does not fall into the wrong hands, nor could it be used as a weapon, for the Axis powers. We want to make sure we do the production here in the US instead. So we're going to have the isolation of a better strain from a cantaloupe. And we're going to be using submerged fermentation. One of the companies that really spearheads this would be Pfizer. Alright, that's just some trivia information on there. And now we're going to be talking about the drugs themselves. Many antibiotics originate from the soil bacteria. So these include the gram-positive bacilli, or the rods, the actinomyces, and fungi. Those are the main sources. Alright, now we're going to go into the characteristics of antimicrobial medications. So what makes an antimicrobial drug effective? So we need this trait of selective toxicity. Selective toxicity refers to how the drug will cause a greater harm to the microbes. Over harming the self. Therefore, we're going to affect structures specific to microbes. Mainly, we're going to be looking at things that are only seen in like prokaryotic cells versus human cells, and then when we have our eukaryotes like yeast or helminths, we're going to also find the differences between those eukaryotes and then a human cell. The toxicity is relative, and it's expressed as the therapeutic index. This is going to be the minimum toxic dose versus the minimum effective dose. Let's highlight some of these. Let me scoot so I'm a little bit in a more comfortable situation. For example, penicillin, didn't mean the highlighter. For example, penicillin G has a high index. If a medication's too toxic, this means we're just going to only use it topically. Antimicrobial action refers to the bacteriostatic chemicals or the bactericidal bacteriocytic chemicals. So bacteriostatic means that it would only inhibit the growth. In bacteriostatic situations, we're going to let our immune system do most of the job and do most of the fighting. Going to let it do the job, so this is going to be usually against gram negatives. Whereas bactericidal, these drugs are going to be just outright killing the microbes, or the bacteria, I should say, to be more specific. We could also talk about the spectrum of activity. So we could have a broad spectrum. When we have a broad spectrum drug, that means we're going to be affecting a wide range. All types of microbes are going to be affected here. These are going to be used for life-threatening diseases. And we aren't going to have any time for ID. So we can't really get a specific picture as of what bacteria is causing it. So then we're just going to use a broad spectrum to treat it. This would be like if there's sepsis. Therefore, when we use these broad spectrums, however, this could also increase chance of the opportunistic infection. An opportunistic bacteria will take over the normal microbiota if we use a broad spectrum drug, and then that would cause an infection since the normal bacteria aren't there anymore. However, with a narrow spectrum, this would only specifically attack a limited range. So this will only target the specific pathogen. You swipe over here. This has the next one, narrow spectrum. Okay. So in this situation, we do need to ID the bacteria, but it's also less disruptive to the microbiota, the normal microbiota. We could also have effects of combination, so we could have antagonistic combination. This is when the medications interfere with each other. We could also have synergistic, when the medications enhance the activity of the other. Or we could also have additive effect. When you take two together, this is the same as adding effects. When you're taking them independently. All right. More characteristics of medications. They need to have tissue distribution, metabolism, and excretion. That's a long title there. So there's going to be, of course, different behavior in the body. And you're going to have to deal with things such as the brain-blood barrier. Only some cross this. So that sometimes could limit what you're able to treat. This is going to be a case in meningitis. If there's a low pH, instability at low pH, let me rewrite that. This needs to be injected. The rate of elimination, or the half-life, also dictates the dosage frequency. A few terms here. Injection, the purpose of injection again. If the drug is unstable at low pH, you need to inject it, and then the half-life will dictate the dose frequency. There's also adverse effects that we need to be worried about. These include allergic reactions and toxicity. This is also going to be the suppression of the microbiota. And of course, this is going to let opportunistic pathogens such as the Clostridium difficile increase. Right, the 5 targets of all antibiotics, the 5 main ones, will be the cell wall, protein synthesis, nucleic acid synthesis, metabolic pathways, and the last one is disruption of the membrane. First one that we'll be looking at be the cell wall. These antibiotics are probably going to target gram-positives, since we're going to be playing around with that peptidoglycan. And these are going to have high, let me go here, they're going to have a high therapeutic index. Penicillins are an example. So are cephalosporins. And these have something called the B-lactam ring. B-lactam ring, let's see. So we have our side chain, a C, an O, an NH, then a C, C, a C, and then in one of those corners we have an N. This area right here is our lactam ring, and it's basically conserved in all of these molecules. Then you'll just have different things on the left side and the right side, but this is that B-lactam ring. So these are going to actually inhibit the enzymes. So I'm actually just going to highlight that. They're going to inhibit the enzymes for cell wall synthesis. These are going to be known as the penicillin binding proteins. Or the PBPs. They're going to inhibit those. The penicillins and cephalosporins are only effective the actively growing cells only. Let's highlight that, actively growing. And then, since they do interfere with synthesis, they're going to stop the peptide bridges between the glycans. And then, of course, this is going to weaken the wall and lead to lysis of the cell. Need to probably highlight penicillin binding proteins. That's a vocab word. We have some differences in activity. One of these differences would be the gram-positives having that exposed peptidoglycan. That's the only kind that will have an exposed peptidoglycan, because remember, gram-negatives have the outer membrane, which will block the antibacterial medications that affect the cell wall. There are also different penicillin binding proteins depending on gram-positive or gram-negativity of the cell. And some bacteria actually make something called B-lactamase. This is, of course, an enzyme, because you could tell by ACE in the name. This is going to break the beta-lactam ring. So again, that's the conserved part of all these cell wall drugs. So if you're able to break that, look what happened. You just got rid of it. There's going to also be extended spectrum. Beta-lactamases known as ESBLs. These are going to inactivate a wide variety of these medications. Then gram-negatives are going to have more of these than gram-positives. Okay, let's talk about the penicillin antibiotics. So this section here is all about penicillin type antibiotics. So we have the natural one. This is going to be coming from the species Penicillium chrysogenum. It's going to be important to highlight. The natural version is narrow, so it only affects a small amount of bacteria, specifically gram-positives and some gram-negatives, which was surprising to me because up until this point we were always taught, oh, penicillin only works on gram-positives. You're going to unfortunately have some penicillin nases from the bacteria. So again, enzyme that breaks down penicillin is a penicillinase. However, the good news is we have these penicillinase resistant drugs. These drugs are going to be developed in response to the S. aureus strains that are resistant, and we're going to have altered PBPs in these Staphylococcus aureus strains. And when that happens, the beta-lactam rings, they cannot bind. And therefore, in order to solve that, we're going to, again, have penicillinase-resistant penicillins. A type of S. aureus that is penicillin-resistant would be methicillin-resistant S. aureus, or MRSA, also known as MRSA. This thing is a nightmare. You're taking, again, a very narrow range drug, and now this strain of Staphylococcus aureus, this MRSA, it's even more resistant to this type of medication that usually would treat it. Okay, so those were narrow spectrum. So now let's talk about broad spectrum penicillins. Let me find where we are. Okay. Broad spectrum penicillins are going to alter the side chains. Remember how we were talking about that beta-lactam ring is actually conserved? Well, here we're going to be just adding things to the side, whatnot. Just changing it as much as we can. Of course, this is going to affect both gram-positive and gram-negative. However, these are going to be inactivated in a greater amount by the beta-lactamases. So that's kind of a downside right there. Then, of course, we have the extended spectrum. In this side of the broad spectrum, there's going to be a greater effect onto Pseudomonas. However, we are going to reduce the activity to those gram-positives because of those beta-lactamases. So these are still going to, unfortunately, be destroyed. We could also have a penicillin added with a beta-lactamase inhibitor. Remember, in this field, it's all about being one step ahead, or trying to be at least one step ahead of the microbes and of the bacteria, but they're, of course, going to be a little bit ahead of us because of all the selection and their resistances that they're just passing down as we're unfortunately selecting them with misuse of antibiotics. However, we could also be clever here and try and combine some of these together. So here we have a penicillin plus an inhibitor of that beta-lactam ACE. That inhibitor is known as clavulanic acid. This is going to, of course, protect the penicillin. Version of this would be augmentin. Alrighty. Let's talk about the cephalosporins. So another category in the cell wall medications. They're going to be resistant to some of them, the beta-lactamases. There's low affinity for gram-positive PBPs, and some chemical modifications are going to have the creation of 4 generations. There's also other B-lactam antibiotics like carbapenems and monobactams. These can't be affected by beta-lactamases. Unlike the cephalosporins, which are resisting some of them, these carbapenems and monobactams resist all. And then the carbapenems are wide range. We have a few more down here. We have vancomycin, which is going to, again, block peptidoglycan synthesis via binding to the peptide side chain of the NAM molecules. They're going to be poorly absorbed in the intestine. So therefore, they need to be administered by IV. That's the only way you could do this. And unfortunately, gram negatives, they're going to stop them from crossing their outer membrane. One last drug we have here is bacitracin. Sorry about that. It's been a long day and a long break. So the toxicity limits it to topical application only. So toxic that you could only use it topically. This is going to stop or inhibit the transport of peptidoglycan precursors, not curses, across the membrane. And this is going to be seen in first aid. Again, a topical use only. All right, let's move on to the protein synthesis medications. When we're talking about the protein synthesis drugs, we are going to be targeting the microbes, or the prokaryotes, I should say, specifically the prokaryote 70S ribosome. Versus the eukaryotes 80S. However, we need to be wary because there are 70S ribosomes in the mitochondria. That's why some of these are toxic. All right. Type here of them that bind to the 30S would be amino glycosides. Sorry, I'm still learning all these names. So these are going to have no effect on anaerobes, enterococci, and strep, because we need active transport. Sometimes you could combine this with a beta-lactam, and then therefore they can enter. We also have the tetracycline. Tetracycline is bacteriostatic. And they can't go into animal cells. I should say animal, not just eukaryotic. And there's a resistance of decreased uptake or increased excretion. And we could use this against certain gram positives and gram negatives. All right, let's talk about the protein synthesis drugs that bind to the 50S subunit. So this is a large subunit. We have macrolides. These are going to stop translation. And this is for those who are allergic to penicillin. This is going to be a gram-positive bacteriostatic. And it's going to be unfortunately blocked by the outer membrane of Enterococci, or Enterobacteriaceae, actually. Alright. There's also chloramphenicol. Chloramphenicol, this is also going to stop translation. And wide range. Alright, now let's talk about the drugs that affect nucleic acid synthesis. So we're going to have fluoroquinolones. These are going to target the DNA gyrase. And the gyrase, again, is the enzyme that breaks and rejoins the DNA to stop, so it could actually uncoil it. This is going to be a wide variety effector. Alright, there's also rifamycins. Rifamycin blocks prokaryotic RNA polymerase. No initiation happens as an effect. This is good against gram-positives, some negatives, and then Mycobacterium. Remember, those are the ones that don't have peptidoglycan. They have this acid-fast cell wall, the Mycobacterium. We also have the metronidazole. I think I spelled that incorrectly. Metronidazole is toxic only to the anaerobics. Because you need to convert it to active form. This is going to bind DNA and results in breaks. Alright, let's talk about the drugs that affect the metabolic pathways. Sorry about that. We're going to focus on the folate inhibitors. Let me scroll down here, sorry about that. These include sulfamides and trimethoprim. So sulfa drugs and trimethoprim. And these are going to stop different steps in the synthesis of folic acid and the coenzyme. So we're actually going to be hitting them with a double feature here. So they're going to not only inhibit folic acid synthesis, they're also going to attack the nucleotide synthesis too. Some of these antibiotics will also affect the membrane itself. And this is going to cause leakage. And this is only topical. The ones here are daptomycin. This is going to go into the cytoplasmic membrane for gram negatives because we could insert it and... Oh wait, shoot. This is for gram positives. Sorry about that. It's used against resistant gram positives. Sorry about that. I really had a brain fart there. Daptomycin is for the gram positives that are resistant because it attacks the cytoplasmic membrane. Polymyxin B will be the one for gram negative. Sorry, I had them mixed up. So B as in negative, so B negative, I guess. All right. Now we could talk about the MIC and the MBC. We got through all the drugs there. All right, so the MBC, not NBC Nightly News with Lester Holt. No, we're talking about the MBC. This is the lowest concentration for the death of 99.9% in vitro. However, the MIC, which is the minimum inhibitory concentration, this is going to be lowest concentration that prevents growth. Still in vitro. I think I spelled that wrong. Yep, I did. Sorry. Okay, so in order for the MIC to be found, we need to use serial dilution. And they're going to be cultures that are added, then incubated, and then of course we're going to find their turbidity. They're going to either be S or susceptible, resistant, or R, or intermediate, which is I. Actually, yeah, SRI, there we go, correct. Correct number there. I mean, not number order. Sorry about that. Oh my goodness. Okay, so, for example, we're going to have these vials, and if these last two on the right are turbid, they're very cloudy, the next most clear one will be the lowest dose needed, or lowest concentration, I should say. Right, the MBC, again, it's just determined by plate count, from the MIC. We could also use the Kirby-Bauer method or the disk diffusion method. This is what the research project for the class my lab partner and I are doing. We're testing it on airborne bacteria around campus, so we're using the disk diffusion method. This is going to be testing susceptibility of the strain against different antibiotics. So this is going to be the measure of the ability of drug to kill bacteria. We're going to have these zones of inhibition, which kind of look like this. So let's say we have our culture there. That's the drug, and then we're going to then get this ring around it, but clearing. Let me make that better. Let's make it larger. Okay, so we're going to get this clearing here, and then we'd measure that clearing, the diameter of it, and then that could be compared to tell if the bacteria was susceptible or resistant or not later on. So again, we're going to get our measurement of the diameter, and we're going to either label them as susceptible, intermediate, or resistant. And then we could also take into account the molecular weight, the stability, and the amount of drug used during this test. We have some unfortunate resistance to antimicrobials that we deal with in our time. And let me write this. Okay, so we're going to either have innate or acquired. So innate resistance is when the mycoplasma bacteria just so happen to have no cell wall, so they resist penicillin. However, we also have bacteria that are acquired resistance. Again, this is going to be when they develop it, either from spontaneous mutation or horizontal gene transfer. Increased use as well as misuse will select for resistant microorganisms. For example, the Staphylococcus resistant to pen G. So here we have the resistance percentage from 3%, and then afterwards all this misuse. Now it's at 99%. Actually, 90 to 99%. There we go. So now 90%, at least 90% of strains are resistant. There's going to be an impact on cost, complications, and outcomes. Treatment. We're going to need to take note of the mechanisms and the way that they're spreading. Some ways that they could have this resistance. Let me see. Yeah, okay. So some ways that we're going to have this resistance are antibiotic inactivating enzymes, such as the penicillinase, we could also alter the target molecule. We do here, we're going to make minor changes to the structure, and Most commonly, this is going to happen towards the PBPs or the ribosomal RNA. That's going to stop those macrolides. And again, preventing binding. That's what these resistant bacteria are going to try to do when they're altering the target molecule. There's going to also be a decreased uptake. So we're going to change the porin proteins of the gram-negative outer wall. So this is only gram-negative. There's also the increased elimination via the efflux pump. So the bacteria may just so happen to have a section in its gene that's going to code for this efflux pump. They make one, and then they just start copying that sequence and then expressing it a ton, and you're going to start removing things at an extraordinary rate where they're just being resistant. And then this is going to, of course, push out the drug. So, spontaneous mutations. I'm going to go back and talk about those. They're going to be low rate during replication. So, one base pair equal the gene, or one single base pair in a gene could create a resistance to streptomycin because it affects that ribo protein. One base pair change, that's all it basically takes. There's also combination therapy. In order to fight this, we use multiple antibiotics at once. So it's unlikely that the cells are resistant to all of these at once. We, of course, also have the horizontal gene transfer, and we're going to be spreading genes. The R plasmid, again, that's the resistance plasmid, small accessory circular DNA. May also originate from soil microbes. We're going to see the resistosome in hospitals. That's a major concern there. We have escape pathogens. These are going to be high in virulence and resistance. This is like our FBI most wanted list in microbiology. So we have Enterococcus, Faecium. We have Staph, of course. We have Klebsiella, Pneumona. We have Acinetobacter. Bomani. Then for P, we have, of course, Pseudomonas aeruginosa. We use that in lab all the time. and then basically any Enterobacter. So those are the escapes. Enterococcus faecium, staph, Klebsiella, Acinetobacter baumannii, Pseudomonas aeruginosa, and then any Enterobacter. We have a fear of emerging resistance. When we discuss these bacteria. For example, the Mycobacterium tuberculosis needs long treatment and combination therapy. There's going to be this multi-drug resistant TB, resists the two first-line drugs. If we're not careful, then we're also going to have this extensive drug-resistant tuberculosis. This one is going to be the next 3 or more second-line drugs or antibiotics. That's what it's going to resist. So you're at least 5 different antibiotics that you're going to be resisting, fortunately. Also have a fear of these Neisseria. I don't know why I wrote an M. Should be an N. Gonorrhea. And there's going to be this new resistance, penicillin. Staph. We already talked about staph. This is going to be resistant to penicillin too. And then now we have PBPs with low affinity for all beta-lactam. Remember, the beta-lactam antibiotics, that's what we're going to be using against the staph because it has a gram-positive cell wall. However, if these PBPs that have such a low affinity, then they aren't even going to bind. These drugs aren't going to bind to these bacteria. And then again, we have our MRSA. Those are like 3 case studies, I guess. So the tuberculosis, Neisseria, gonorrhea, and Staphylococcus aureus with MRSA. There's concerns about overuse, wide availability, and animal feeds. Those are like 3 main concerns with antimicrobial medications. For example, there's Salmonella. That is now resistant to antibiotics due to the usage of antibiotics in animal feeds. Alright, mechanisms of action against viruses. This is our last stop here. So they're going to be more difficult to target specifically. And so we're going to need to make the antivirals, which will stop replication all these parts. So viral entry, uncoding, the dogma basically, DNA to RNA to protein, integration. Oops, there we go. And release of the new viral particles. there's only going to be specificity against one type. There isn't like a magic cure for every single virus. Unfortunately, we're going to need to develop an antiviral specifically for that one. With COVID-19, which as of recording this video, there's still cases of it going around even though it's November 28th, 2023. And so it's just, again, best to stay safe and stuff. When my friends, his family had COVID recently in the past week or so. So some antivirals against COVID-19 include Paxlovid. This stops the protease. Remdesivir. This stops RNA dependent polymerase. And the molnupiravir. This is going to mutate the RNA. And then you might be thinking, mutating RNA, isn't that causing all these variants? No, this is going to introduce harmful mutations to the virus particles. All right, that's going to be it. Those are all the antimicrobial drugs for that chapter that I need to memorize and study for the next test. Thanks so much, everyone, for tuning in. Please do something nice for someone, and good night to all of you.